Okay, thank you very much. I'm very happy to be here and also to have my very first opportunity to be in Taiwan, even though it's for only 36 hours. I have to fly out tonight. I have the first time. Okay. This is the rather nice, where did it go? This is the rather nice city of Perth on the Swan River, which I visit about 1 month every year. Can something be done to prevent the city of Perth from disappearing? Which I visit about 1 month every year to do my duties in the University of Western Australia. This is the Swan River, which, like all major Australian cities, Perth is situated on a river, and like most of Australian cities, it is named after either a British city or a British aristocrat, something which I personally find rather regrettable. What's the matter with my slides? Why do they keep disappearing? They're not that bad, really. I mean,
forward is the right-hand side part.
Yes, but that's not the problem. The problem is they keep disappearing. Okay, well anyway, let's move on. Okay. So this is the outline of my talk. I'm going to look at some general principles, and then I'll go over several different frameworks. I deliberately did not include Japan because I knew that it would be covered in this symposium. It's an important framework. It's a very interesting framework. Also, Taiwan will be covered. So I'll look at the US approach. We all, as in everything in life, have to take into account of what the United States does. I'll look at the European Union, and I will round off with Australia, which is my country of origin and which is a framework which I personally influenced very strongly when I was in the Therapeutic Goods Administration. From all these different international approaches, I will draw several discussion points and lessons which I think will allow me to then move on to the conclusions. I have to say that what you have in your booklet is different from what I'm going to present. There are a little few less slides in what I'm going to present. There are also a couple which are not in the booklet. So anybody who wants these slides, just ask the organizers or ask me for them. And the bibliography of the talk is basically embedded in the various studies which are used to illustrate the slides. Now, we've just had a bit of a flurry between Professor Shimashad, Mickey, and myself on the question of how to define cellular and tissue therapies. Basically, we have in every regulatory system in the world a drug-device divide because there are obvious differences in the way medicines and medical devices are manufactured and presented, and therefore it has been a reasonable approach to develop different definitions and sometimes different legislative provisions. Drug and device principles intervene and interface in relation to the generation of tissue and cellular therapies. In each and every framework which we have seen, this was very evident in the Japanese framework, it will be evident in the ones I show you, certain aspects of cell and tissue therapy type interventions are viewed as being more in the realm of medical practice than in the realm of manufacturing practice. This has been acknowledged throughout by regulatory authorities, and exemptions have been generated at the very outset of the development of frameworks. I shall discuss these exemptions, and I shall show how experience has taught agencies to be somewhat cautious in the way in which they develop and allow exemptions, with particular reference to the very interesting example of Australia. I think I will go very quickly over many of these general principle slides because this is obviously an expert audience and I don't have too much time. This is basically a cartonic representation of why, in fact, we do treat the cell and tissue therapies somewhat differently from the mainstream pharmaceuticals of large-scale batch manufacture. And you can see over here the characteristics of each type, and you can see that it is indeed the case that given that we are, as has been said, looking at single product, low throughput type products with a high level of labor intensivity compared to established pharmaceuticals, with a limited control on the starting material compared to bulk raw material, and with a sector which is evolving from the research environment embedded in medical research and so on, we can see that these do deserve special consideration. This is just a slide which I thought looked rather nice, so I thought I'd put it in, but it just shows the kind of complex interventions which are involved in the so-called manufacturing of cell and tissue products, and the ways in which both medical and medicine and device type applications can intervene and be relevant to these products in terms of how they are being manufactured in a cell therapy process. So I'm now going to talk to you about the international approaches. This is my charge. And I'll go over several aspects here. This is pretty much a sort of personal preference type summary of how I view this sector. First of all, there's the management of so-called TERP issues, territory within regulatory agencies. Like all human organizations, regulatory agencies are bureaucracies which are made up of individuals who are very, I would say, interested and jealous of making sure that their own interests themselves are served within the agency. And this is something which is very visible in the evolution of cellular and tissue therapies. When deciding which part of the agency actually deals with the regulation, that is the first hurdle which manufacturers have to overcome. And I'll say something about that. A very, very common and in fact universally applicable aspect of cell and tissue regulation worldwide has been the so-called tiered regulatory approach. You heard that the Japanese system is based on classes. You will see that in some shape or form all systems are based on classes, including the Australian one. You will see, you will note, I noted with some level of amusement that the Japanese have classes 1, 2, and 3. So do the Australians, but they invert the way in which they actually address them. There is a very big interest in infectious disease minimization and the prevention of contamination. This is the basis of so-called good tissue practice. Yesterday we were interested to see a GTP lab at the university. After our meetings, it was very interesting to see and to assess the level of compliance on a cursory basis. And then there is, of course, the assumption that given that efficacy is so difficult, the first models, the first attempts to regulate this sector have been emphatically focused on manufacturing assessment rather than efficacy and therapeutic claim assessment. But this is something which has changed very much, and I think it's very desirable that it does change. This is just an example, ladies and gentlemen, of what I meant by the management of TERP issues. It is my understanding that the Presidents of the United States and the President of North Korea signed a memorandum of understanding of 2 pages long in relation to matters of nuclear war and relationships and peace in the world. That was 2 pages long. This document is the intra-agency agreement between the Centers of Radiology and Device and Radiological Health in the FDA and the Centers for Biologics Evaluation and Research. This agreement was signed in 2003. It is 12 pages long, and you can get it on the internet. It details down to the last comma who does what, and God help you if you breach the agreement. I suspect nuclear war may well result. As I said, one of the basic principles is the tiered regulatory approach, and this is just, and I will be coming back to this principle time and time again, so I will not belabor it. This is the way in which the US FDA has addressed the issue. It is particular to the FDA because it is a reflection of their particular legislation, which I will outline briefly very soon. But basically, the principle is that as we approach a higher risk-to-benefit ratio in these devices, in these biologics, in these cell and tissue therapies, we will see a different and higher level of regulatory oversight kicking in. This is a basic summary of the phases of regulatory and clinical development. The preclinical, the CMC, the manufacturing and the manufacturing control stage, and the clinical aspects. All of these must develop together. They are not independent, but it is true that in relation to cell therapies, they are very different from mainstream pharmaceutical development. For example, if we look at the concept of clinical trials, I do not think that the concept of phase 1 studies has any relevance at all to cell therapies. There are hardly any that I know of phase 1 studies, which means safety studies in normal volunteers. These therapies are developed and pretty much immediately applied to the patient population. So this presents reflection and some levels of challenge. These are the concerns which are, in my view, unique to cellular products. The source of the product is obviously very unique, and other aspects such as the batch size, the timing, and so on. But it must be accepted, I think, by the sector that every sector thinks that it is unique. I myself have a strong background in the blood sector, which is how I eventually got into cell and tissue therapies. And I have always been conscious that the blood sector itself has always claimed to be unique, claimed that it needs exceptions, claimed that it reflects urgent medical need, and claimed that it reflects the public good and is, in most Western societies, funded by the public purse. These are reasonably close to the arguments posed by many cellular therapies. At the same time, the blood sector, like the cellular therapy sector, is commercially a very vibrant and important sector, and is not isolated from the rest of therapeutic reality. I will just make some points about the issue of clinical study, because I think that we are agreed now that today, in 2018, our focus on cell therapies is how to do clinical studies and how to get them to the stage that they can be shown to support a therapeutic claim. And I think that one of the most important challenges lies in what now we accept in therapeutics, which is the difference between small-scale, efficacy-based studies and what happens then once the product is released in the general population. I think that this is something which is very important, and it is focused by the question of what happens when a more heterogeneous population of patients with the disease are exposed to the treatment. We all know about the question of manipulation. I'll come to this more and more times, but this has been one of the differential factors, if you like, in relation to getting aligned to the level of risk-benefit. And this is basically a very general way of showing the manipulation concept as universally or semi-universally applied. I shall show some definitions from the different agencies in a second. I thought it was interesting to show this slide showing how the sector has evolved. And it is of interest to me that there has been a remarkable shift from autologous to allogeneic type applications. I hasten to add that I do not concede, and I shall show this as well later on, that an autologous application means that it does not have to be regulated, or that it has to be regulated in, let us say, a less strict way. I think that a patient who receives an autologous treatment has as much rights as a patient who receives an allogeneic treatment, and there are risks there as well. But it is interesting that the autologous sector is on a relative scale, given that the number of therapies is increasing considerably and becoming less visible compared to the allogeneic. Also, the disease targets have shifted considerably in the past 20 years or so. And this is just a cartonic representation of the number of treatments which were, at least with a market approval when this review was published in Biologicals. It is still a modest number compared to the vast number of therapies which are under trial, being developed, or under market review by the various agencies. This is the situation in the countries which you can see here. Now, I was told to look at international perspectives. I'm going to draw your attention to this very, very good resource provided by the Japanese agency, which is the proceedings. I suspect that many of you in this audience were at this symposium, which was carried out in Japan a couple of years ago. You have the reference there in your slides, in your presentation, and I do recommend that you use this as a resource if you want to find out what the peer agencies are thinking about at this point in time. Let's go to the Americans. As I said, very important. So the United States, cell therapies, gene therapies, tissue engineered products, all these will come into the definition, which is actually relatively new now, because the FDA has suddenly, as of the late last year, started to call these regenerative medicine products. And these are regulated either by the Center for Biologics Evaluation and Research or the Center for Device and Radiological Health, as I outlined before, under the auspices of 12 pages of incomprehensible bureaucracy. You can see here the framework under which these are done in relation to the definitional aspects and in relation to the who does what aspects. The FDA's risk-based regulatory framework. Nowadays, it is very fashionable for all regulatory agencies to talk about risk-based regulation. As a sort of old ex-regulator myself, I am somewhat puzzled by this, because as far as I know, we always practiced risk-based regulation. We always were very mindful of risks. But basically, I think that this is pretty much a reflection of recognition of issues in the environment, including resource issues for the regulators and industry pressure in terms of trying to get products on the market. In the United States, the US FDA is somewhat unique in terms of its being subject not to one, but to two regulatory laws. Prior to the introduction of the Food, Drugs and Cosmetics Act, which is the main regulatory law overseeing the activity of the Food and Drug Administration, the Public Health and Safety Act of the United States was actually in play already under so-called Section 361 to oversee the public health in relation to issues such as I've mentioned before, infectious disease minimization. So when the FDA was still in a position that did not have the powers to actually oversee much of tissue and cell products, it was drawing substantially on 361. And in this aspect, it was looking at comparatively simple products such as banked tissues, which were subject to the aspects which you see over here. I wonder which one of these is the pointer. Okay. The products which were minimally manipulated only for homologous use. All these definitions we will see very soon, but I think they are familiar to you. And these were not requiring pre-market approval and clinical studies. They had to comply with good tissue practices under this part of the Code of Federal Regulations, and these good tissue practices were firmly focused on infectious disease minimization and the avoidance of contamination. The high-risk products, which came later, which were more novel and more complex and had high-risk applications, were then regulated under 351 of the CFR, which included good manufacturing practices and all the kind of provisions involving preclinical assessment, clinical trials, and all the other aspects of mainstream therapeutic regulation in the United States and internationally. This is a somewhat complex but quite useful algorithm of what actually happens in the United States. I will not belabor it too extensively, except to say that products are taken figuratively, if you like, through this algorithm to decide whether the various criteria for 361 products are applicable, whether they are minimally manipulated, homologous in use, in combination, etc. And ultimately, in a fairly straightforward fashion, you get to the conclusion as to which part of the law and the regulations you can actually use to apply this. It has to be emphasized that in the United States, all the powers of what is regulated lie within the FDA. There are no, if you like, delegation outside the mainstream regulatory agency. such as is found in many other countries. We've heard an example in Japan with this plethora of committees, as well as in some instances even in Australia. These are the requirements which you can see here in relation to good manufacturing practices, good tissue practices, and good clinical practices. These are all fairly standard, especially the GMPs and the GCPs in relation to therapeutics regulation generally. The FDA, like I would say my agency in Australia and perhaps most agencies, has been reluctant, has had to be dragged somewhat reluctantly to the concept of developing separate frameworks, and has continued to rely both in relation to the instruments used to regulate, as well as in relation, as I shall show, in the instruments used to facilitate access to the agencies on traditional established instruments which are in place for both medicines and devices. Now, it is interesting, as I said, and I think this is the result of an accumulation of, let's say, experience as well as problems after the actual introduction of this framework over the 2000s, that the FDA late last year issued 2 final and 2 draft guidances in what it called the Oversight of Regenerative Medicine. As I said, now they are calling these products under the umbrella of regenerative medicine, like most people. And I have studied with interest, excuse me while I have a drink. I have studied with interest these guidances, and we don't have the time to review. What do I do? Should just press enter or what? Okay, it's good. Thank goodness. I have studied these guidances, and I do recommend that anybody in the sector studies them, because irrespective of whether you want to market in the United States, they contain a lot of concepts which I think are notable, and I have, for your benefit, extracted those which I think are most notable. And in relation to the guidance on minimal manipulation and homologous use, these are just the definitions, which you can see here. The definition of minimal manipulation, something which is used now quite widely internationally in relation to this sector, and the principle of homologous use. This is a very important concept. I will not go over this, just reading it, I leave it to you, but this is basically pretty much what most people are doing. I don't seem to be getting the slides. The definition of same surgical procedure. As I shall describe later on, this has been a very interesting issue in which the intent of the regulations internationally, certainly this has happened in Australia, has been, so to speak, overtaken by an interpretation which could be allowed under the law, which has allowed some parts of the industry to be, let us say, somewhat cavalier with the intent. I want to make this point. As a former bureaucrat, needing to be always aligned to the relative law passed by the parliament of my country, it was emphasized to me by the lawyers in my division in the Australian government that the law basically gives out an intent. There is a difference between intent and implementation. It is very important to recognize the intent. It is unfortunately the case that people covered by the law very frequently manage to find loopholes with the law, within the law, which are not covered by the intent. And this has happened, as I shall show you, in cellular therapy regulation. Next slide. Okay, this is the draft guidance on the question of expedited programs. Read this guideline, because it will show you what kind of measures you should suggest to your respective countries in relation to the special areas in expediting, in making faster access through the regulatory agency, and special provisions in regards to issues like patient size and population and so on. This is quite important. But what is also notable in this guidance is the definitions. Now we have a definition within the Food, Drugs and Cosmetics Act in relation to what are regenerative medicine therapies, which are what were formerly called HCTPs, and that an investigational drug is eligible for the designation if it meets the criteria which you see down there. Next slide. It is very interesting to me that the point which I plugged out in relation to the issue of the devices used in combination therapies with regenerative medicine products was the very interesting concept, and I would welcome comment on this, that the agency does not consider scaffolds combined with the cellular product to be within the scope of the guidance. And there is the reason for this. I was intrigued when I read this early this year, because I can remember very big territorial fights within the TGA between myself and the head of the devices and biomaterials division of the agencies as to whether scaffolds within the context of a cellular therapy were a device or were actually part of the therapy. I am glad to see that the Americans, for once, have done something with which I agree. Next slide. Okay, this has already been somewhat cursorily shown, but this is just the development of the clinical development of any therapeutic, including the cell and tissue products, or as we must call them, the RM products. And I will just make a couple of comments. It has been commented by many authorities, including our eminent colleague from Japan, that preclinical models for cell and therapy products are actually quite difficult. I would agree that this is the case, but I would also suspect that science is developing these pretty much at an accelerated pace, and I think that there needs to be some level of higher focus on these relative to the clinical phase if we are to assure that basic safety issues are addressed before these are put into patients. But it is true that this is a problem. I've already made the point that I think that phase 1 studies are probably not at all relevant for RM-type products, and possibly the classical division between phase 2 and phase 3 is also quite difficult given what we mean by phase 3 and how can one, execute a phase 3 study with these products when most of them are submitted to a very specialist, low number of centers. Can I have the next one? Okay, let's go now to the European Union. Well, the European Union, ladies and gentlemen, is in a mess at the moment, as I suspect any of you who are looking at the international situation are aware. Pretty soon we will be sadly saying goodbye to our British colleagues who have declared, incidentally, at the level of regulation that they would like very much to stick to the European framework. They've lost the European Medicines Agency in London, along with quite a lot of money. It's gone to the Dutch, which means that they'll have to go to Amsterdam instead of London periodically. But the point is that the European Union, like in most other things in relation to this sector, has a substantially complex legal and regulatory framework, which I have tried to show on this summary is actually, like with the FDA, drawing on other parts of the European legislation along with the specialized aspects which have been, as I said, slowly developed for cell and tissue products. So the location of regulatory oversight, in this instance, the centralized procedure, that is something from the past. The whole question of GMPs, that has already been established. The pediatric rule, very relevant to cell therapies in relation to young patients, that has been there for some time. But what is relatively recent over the past 12 years or so is the concept of the advanced medicinal products, which was developed and given its own oversight and committee as a result of new legislation. Can I have the next slide? Okay, these are the legal texts of relevance. Some of them have been shown on the previous slide, but I found this slide quite useful myself because it shows an important aspect in all facets of European Union law, which is legislation which is applicable, imposed, if you like, centrally across all of the member states, whether you like it or not, and legislation which is delegated to the individual member states because it is considered to be more particular in terms of individual health systems in this instance, in terms of individual funding models, and so on, to the actual member state. So you can see this, that there is national implementation for several of these kinds of legislation. Next slide, please. Okay, so let's go to the ATMPs, the Advanced Therapy Medicinal Products. As I said, these cover a number of products, which you see over here. They are reasonably analogous to the 351 products of the FDA, and you can see them summarized here. And the Committee for Advanced Therapy Products is the one which actually oversees them and develops the recommendations for eventual approval and ratification. Next slide. This shows the process. It is, as I said, for these products, a totally centralized procedure. These are evaluated on the basis of the provisions which you see over there, so they are reasonably analogous to the FDA's 351 process involving clinical studies, preclinical, and all aspects of manufacturing oversight, including GMP, and they are centrally overseen by the European Medicines Agency, with the Committee for Advanced Medicinal Products developing its recommendations to that agency, and then ratified by the European Commission. Next slide, please. And this is just a summary, reasonably current. I think it is as of the middle of last year, of what we have at the moment in Europe. A reasonable number of products in the various different categories, and some interesting comments down there. I've already mentioned Glivira, the sole gene therapy which was withdrawn. Provenge, we have heard about the somewhat sad story of its commercial demise. Next slide. And this is the relevant framework or the relevant guidances applicable for genetically modified cells. So as you can see, there are quite a number of legislative instruments. I don't have the time to go into this in detail. I recommend that you actually have a look at the different ones which are extractable from the reference on the EMEA website down there. Next slide. Australia, a country which is as close to Taiwan as it can get. It only took me 9 hours to fly there. When we started to look on these, and now I come into the realm of personal experience and perhaps more importantly, personal prejudice. When we started to look at these in the late 1990s, about 20 years ago, we basically developed a definition which pretty much captured everything. And we decided that the only area which could be definable as cell and tissue therapy was the area of blood, and this was for the simple reason that we were already regulating blood with its own framework. It was a somewhat sensitive area, and it had sufficient differences from the mainstream treatments of cell and tissue manipulation that we thought a different framework was required. So I'm going to be using the word biologicals as a catch-all for HCTPs and the kind of products which are available everywhere, some of which are not regulated to the same level as highly developed cellular therapies. The framework was introduced in 2011. It was developed over a considerably longer period of time. I think the first time I met Mickey was in Singapore. I think it was in the early 2000s when we were, as an agency with a relationship with the Health Sciences Authority, developing this framework, and we were interested in discussing other people's experience. It took an extremely long time, a hugely onerous process of developing consultation, inputs, modifying the framework, introducing legislation, and so on. Okay, I didn't ask, but here is the framework as it currently stands, and you can see that most of the products definable as biologicals are regulated as such under the biologicals regulatory framework, which I shall describe. There are some products which are excluded. I will specify what these are, and I will update you on this important aspect. And there are other products which are regulated, covered by the definition, but not as biologicals. Next slide. Okay. We have, as I said, similarly to Japan, a system based upon classes. The logic of the classes is exactly the inverse of Japan. In Japan, the highest level of development and sophistication is class 1. In Australia, it is the lowest. Only, of course, we don't have class 1 anymore. We used to have class 1. I'll come to that in another slide, but I might as well tell you now. We had class 1, but we had to drop it, because after its introduction, there was such a huge groundswell of opposition that the TGA, being a bureaucracy responsive to politicians, the politicians in their usual, let's say, flexible, a word which I heard Dr. O say earlier, in their flexible approach decided to drop class 1. I would say in their craven approach, and in the fullness of time, I'm sure they will regret it. So we have class 2, class 3, and class 4. And basically, as you can see over here, as the level of manipulation and the intervention of non-homologous use comes in, and other levels of sophistication such as genetic modification comes in, the class number increases. So these are some of the examples that you can see on the slide. MSCs come into class 1 when they are not substantially manipulated, and when you get genetic modification and other forms of transformation, you go into class 4. Next slide. This is my own cartonic representation from many years ago. I have a sentimental fondness for this slide, but I kept it because, as I said, class 1, which was previously involving cell and tissue therapies captured by the definition and composed of unbanked and unprocessed materials such as whole solid organs. We also had reproductive tissues in this slide, in this class. We also had things like unmanipulated bone marrow, and these were all captured as class 1, and they were intended to be regulated purely through assessment of infectious disease risk and contamination type risks. As I said, after industry pressure, and when I say industry, I don't mean high-level multinationals, I simply mean the sector involved, which in this instance involved mostly public hospitals. I'm told that I'm approaching my end of time, so let's go on. Okay, next slide. Okay, this is basically showing the gradation from medical to manufacturing practice, which I have tried to emphasize. Next slide. Now, we had exclusion, and I want to spend some time on this. The excluded goods are covered over here, and you can see that what we've got here is the intention to exclude purely medical practice of relatively unmanipulated procedures done within the same hospital. Unfortunately, as I discovered after I left the government, they had good cause to regret this. Let's go to the next slide. A number of publications started to arise which indicated that problems were coming out as a result of this exemption. This is a publication from some Australian authors which outlines this. I will now show you the specific example which caused the media to be interested. Next slide. Next slide. Okay. This is a clinic in Sydney which is extremely entrepreneurial, and this is their medical director, and I am simply here because I don't want to end up in some kind of litigation, showing you an extract from the ABC, the Australian Broadcasting Corporation, some years ago, just 4 years ago. And next slide. This is a debate coming again from the same program regarding the activities of this and similar clinics in Australia. And the debate, interestingly, drew in the very eminent Professor Itescu, who in Australia oversees an agency which most of you are aware of, a company which most of you are aware of, which is extremely committed to develop cellular therapies with massive investment compared to this clinic. Next slide. Next slide. This is the incident which caused the public to become extremely interested in these activities. A woman died after undergoing a liposuction procedure which was to treat her dementia. Okay, the MSCs were supposed to, well extracted because of the conviction that using these MSCs you could address dementia. Next slide. And this is more of the same in relation to the public and academic comment about this. I don't have time to go over this in more detail. Next slide. And therefore, this resulted in proposed changes to the Therapeutic Goods Act in regards to the regulation of autologous cell and tissue products, so that now the intent is having to be further refined by ensuring that commercial activity, which is actually resulting in non-homologous use and which is being done outside the mainstream medical environment, is the area which is regulated. And I say this because it is all very well to talk about autologous, but when you are using mesenchymal stem cells as an autologous treatment for dementia with absolutely no evidence base, I think that a regulatory agency has the imperative to act, especially when safety issues are clearly at bay. And the whole question of within the same hospital, which you will remember came up in the FDA's definition, demands to be addressed. What is the definition of a hospital? Is it the clinical area where practice actually goes on, or is it a laboratory in the basement of the hospital where the cells are very quickly whipped down to be manipulated? This is what I mean when I say the intent of the law has to be respected, and if the same efforts were to be done on respecting the intent of the law as are done in dodging the law, I think we would not have the kind of problems which we've seen here. So these regulatory changes are, I understand, underway. This is the situation in Europe. You can see here in Italy, Germany, and Hungary, we've had similar types of incidents arising. And I think that this is giving regulators, let's use a word which is controversial in Europe now, a level of cynicism in relation to some of the procedures which are going on, and the determination to rank up the regulatory system. Next slide. Next slide. Let's skip this one. Next one. And these are the kind of things which I think are needed to counter this kind of incident, premature commercialization, and the regulation of non-evidence-based stem cell therapies. And I emphasize the words evidence-based. All of medicine these days is based on evidence-based medicine, and I do not accept that this sector should consider itself to be exempt from the need to acquire clinical evidence. These are some ways. I do want to emphasize that there are ways in the clinical development which I think the sector and regulators need to take into account and become familiar in order to develop more innovative clinical development pathways. These are some of them here: Bayesian analysis, N of 1 trials, sequential analysis. Next slide. I developed these in a paper some time ago. You can go and look for it if you like, but there are many other more recent and better publications. And the point I seek to emphasize is that both the European Medicines Agency and the US FDA have committed to using these kinds of approaches. They have so far used them only for mainstream pharmaceutical agents, but it is up to the sector to show that these are applicable. And if this is shown, it will be shown that many of the problems in relation to clinical development can be overcome. I believe I'm towards the end. Next slide. Good. So these are basically my conclusions. I think that in terms of what I've been charged to talk to you about, the common features of international regulatory systems, we have the established principle that the requirements are proportionate to the risks. This is, I think, very well established now for all aspects of therapeutics. we still have this ongoing debate as to what is manufacturing and what is medical. As I've said before, I think that this debate needs to reflect the intent of regulation and the intent of the law. We know that we have tiered approaches and requirements for GMP and GCPs when these are applicable to the higher level products. I'm very happy to have heard about the need for harmonization. We need it. It is still in its infancy. And yesterday I learned that Taiwan is now in the ICH, and I do encourage the Taiwanese agency to advocate strongly for more ICH involvement in cellular therapies. I think that the management of most cell therapies will be the management of clinical trials. The sector is now quite used to the fact that it needs to spend a lot of money in generating good practice labs, in generating GMP, in making sure that the products are manufactured in a controlled environment. It needs to accept that it has to develop the same commitment to establishing clinical efficacy. And therefore, for this, I think we need to have innovative designs, as I have said, for clinical studies. I apologize if I've gone over time, and I thank you very much for your attention.
We're a little bit over time, but I think maybe one short question is still okay. Any question? Well, if not, and
can I just ask one quick question? Sorry. Just one. Albert, do you want to just quickly comment on some of these cell therapies in some countries, again, not bypass, but you use the either hospital exemption, compassionate use, or that, you know, that because this is a life-saving measure for someone with a rare disease or with no other options available, you don't need to do a clinical trial and you could do this. Can you just comment a little on this from the regulatory point of view and what you think about
Well,
this?
look, this is quite an important issue. First of all, let me say that the sector, without wishing to be patronizing, needs to not be sanctimonious. This is not the only sector which develops treatments for conditions for which there is a desperate medical need and for which there needs to be an understanding of the relevant conditions. Okay? Secondly, certainly in my country, and I suspect in all others, there are already provisions which allow this to happen. There is only one aspect in some of these provisions, which is the buck has to stop somewhere. And when this happens, the buck stops with the relevant providers. Okay? So this is, I think, very important. What is, I think, equally important is the realization that if you're captured by a law, you should spend more energy on coming under the law than in dodging the law. And I have to say that I've been out of the agency now for 10 years, but I'm a battered individual who had to counter much of my energy in relation to people asking me as the first question, not what are the requirements, but how do I get out of doing this? I think that if people raise all this money in order to develop these treatments, then they should comply with the requirements. But, you know, maybe we could discuss this ad infinitum. I don't think this is ever going to be resolved.
Thank you, Professor Farrugia, for your informative speech on the development on the regulatory front. Thank you.